Thanks very much, Craig. I'm going to get right into things here in the interest of saving up some time, and I think we're all gathered here today because we do recognize that there's a really exciting opportunity in forest biomass, and this first slide just sort of outlines some of that opportunity. The point that I'm going to make relevant to my presentation today is that one of the barriers we face going forward for forest biomass, and Murray has alluded to this, is having a suitable inventory designed to actually look at some of the new traits that we're interested in our forests. One of the most exciting developments that I see is if we look at the inventory and its development through history, for about the past 140 years we've been focused on looking at merchantable saw logs and pulp. But there is a lot of new technology being brought forward in an inventory sense that can allow us to do some new things. Being an ecologist, I'm interested in the fact that previous inventories, the polygons in the inventory are usually defined by species composition, but inside those polygons, out in the fields, you can find that the environmental conditions actually change quite a bit. And there's a move forward now to actually develop inventories where the polygons are real ecosites. And an ecosite is a combination of vegetation and soil characteristics that has a lot of meaning in terms of forest growth. So it's exciting to be able to look at that. Another exciting development is through some of the modeling efforts that Joe and Murray have talked about, we're able to look at delving at finding new attributes for the inventory. So one could think long-term of a grand vision of a forest system, a system like a green pages that actually would be a way to quantify where and exactly how much and what the quality of different forest products are. And these forest products could represent a range of values, traditional and non-traditional. And today I'll just focus again on this idea that this inventory could include something like biomass. And this is an idea that has been around and of interest to tropical forests for some time that was originally proposed by Daniel Jansen. So what would a system like this for our part of the world look like? Here's a blueprint of just a schematic, conceptual model of how you could develop such a system, and it involves a lot of the things that you've already heard about today. I'm just going to make 2 points, and you can have a look at this presentation on the web later to see more detail. But the first thing is that it should be built to fit into our the available standard forest resource inventory data. And the second thing is that we should be looking at what attributes of interest we'd like to be able to model from those standard data to expand or enhance the inventory. And the key here, the key to our work here at Nipissing is that we're looking at developing predictive models where we take an attribute of interest and we look for a mechanistic relationship. So what environmental data can we bring to bear to tell us how much biomass of black spruce will grow in this particular stand, let's say. And so we use those environmental data which come from sources like the Forest Resource Inventory and digital elevation models, etc. So the main objective of this project then is to help develop some of those tools that could be used to enhance the inventory and add new attributes and base those attributes on ecological relationships. And the 2 key innovations I'll talk about today are ecologically based growth and yield models, so looking at how environmental conditions affect growth directly. And then secondly, hopefully I'll have time at the end to just briefly discuss some new work we're starting with terrestrial LiDAR and its use in defining exactly what products might be available from a tree. So very quickly then, I'm just going to focus today on some work we're doing in the Romeo Millette Forest near Timmins, Ontario, and we're looking at 3 major boreal species, black spruce, jack pine, and trembling aspen, and I'll just talk about black spruce today. And I'm going to first of all outline an approach that we're looking at for ecological growth and yield that looks at permanent growth plots and also looks at dendrochronology. So Ontario has a network of permanent growth plots. These are 11.28 meter radius fixed area plots that are designed to represent a specific forest condition. They're remeasured every 5 years, and in the Romeo Millette there's approximately 130 of these. So one measurement or 5 years of data are available for this forest at this time. So we acquired these data from the Forest Ecosystem Science Co-op, and we've done some initial analyses on these. These are some preliminary results that I'm just showing as we work through the data. What we wanted to do was look at diameter growth of co-dominant black spruce. These are the average trees in the stand that are not shaded by other trees but are competing well. We calculated the diameter increment. That's basically the difference in diameter for 2 trees between time 1 or time 0 and 5 years later. And because we're interested in biomass, we took those calculated diameter increments and we used an allometric model like Joe discussed today to convert those into above ground biomass estimates. So if we take a look at, it's a little bit dark to see this, but if you take a look at this graph, this is a first initial run at looking at diameter increment across a range of ecosites. And those ecosites are part of the new Ontario Ecological Land Classification System. So boreal ecosite 34 is a dry sandy ecosite, and this is a gradient going to organic wet ecosites. 2 points I want to make here. The first is that at the ends of the gradient, the drier and the moister ends, diameter growth was lower, and at the middle of the gradient, the mesic part of the gradient, we noticed that diameter growth was higher. If we then take the average tree that would have been growing in these different ecosites, because we don't want to confound our results by including different size trees, so we just calculated the average tree, and we asked the question, if we add that average increment for each ecosite, how much biomass difference would that be? So this graph is the same, except it's biomass increment across that gradient of ecosites. I'll just draw your attention to 55 and 128. The diameter increment in 128 resulted, and this is per tree, of a biomass increase over 5 years of about 8 kilograms. If you look at boreal ecosite 55, it was 18 kilograms. So at a tree level, anyways, we're able to show that these data suggest that ecosite really does influence the production of biomass. But these are only 5 years worth of data, and we would like to have a longer record to look at these growth differences. So we've also chose to take a dendrochronological approach to the problem. We can do this because tree ring records have been shown to give good representative growth trends when you relate them to permanent growth plot networks. And in fact, they're in some ways better because you can get the data for 100 years of growth in about 15 minutes instead of 100 years. And on top of that, you can get longer growth records and you get annual data. We based this method on a model proposed by Cook in 1987, who basically said that tree rings are information, and this information can be deconstructed for different purposes. So our whole goal is to deconstruct the information so we can say something about different ecosites. So here are two plots showing about 80 years of growth for a plot that had the similar ages and similar basal areas, but different ecosite. 128 is the red trace, the wetter ecosite, and 44 here, the blue trace, is a drier ecosite. They have very similar growth trends over time, but can we take the ring width along these series and can we decompose it into different signals? So if we use Cook's model, the first trend that you see in these data is that trees, as they get older, they grow slower, so the ring widths decrease, and that's also a result of the geometry of the stem of the tree, that as it gets wider, you add less wood to the stem. So there's a negative age trend in both of those signals. From year to year, there are ups and downs that relate to good or bad growth, likely due to climate, precipitation, and also possibly temperature. You do see across the whole Romeo Millet sometimes a dip in growth that trees all over the landscape show. These are widespread disturbances, and we suspect that this one is related to a spruce budworm outbreak that occurred in eastern Canada about this time. Again, these are just single plots, but we're exploring this. Within any given series of trees, you can have the average for the site and one tree that's behaving very differently. It grows slowly to start, and then for some reason, it grows very much faster than the rest of the trees in the plot. We think this is because, again, probably a tree was knocked down next to this one, gave it a bit more light, and it grew much faster. But if you take all of those signals and you filter them out, what you're left with is a growth difference between two different ecosites, and that's the signal that we're interested in developing and looking at. So the way that we do this is by replicating and stratifying. So we take several samples of trees from every plot, and we take plots from the same age class and the same basal area, and what we vary is the ecosite. And then we look at growth between the differences between those two ecosites. I'm going to really slip through the methods quickly here, and if you want to read up more, again, you can have a look at this on the website. But basically, we sampled trees around the permanent growth plots in the Romeo. We sampled 8 trees per plot, and we selected these trees at random. We measured DBH height, and we extracted a core from 1.3 meters. And we used standard dendrochronological techniques to surface the cores, and then finally to create an index of ring widths over time. So what can you get from this index of ring widths? Using models, you can reconstruct first diameter growth, and again, these are the two plots that I showed you previously. So this is reconstructed diameter growth based on tree rings. And use a simple formula there of basically just taking the radius and multiplying it by 2 and adding it to the previous year's diameter. I won't go through basal area, but what's interesting here is you can also, if you know the reconstructed diameter, reconstruct total above ground biomass. And so here's a graph that shows over time the accumulation of biomass of a single tree in 2 different ecosites. And so if we develop that a little further, and we take samples from different ecosites that are the same age and the same environment, you can see that there are differences in the growth trends, the radial growth over time, and these translate into fairly big differences in biomass. And in this case, the drier site was producing much more biomass than boreal ecosite 65 or 128, the wetter sites, to the tune of almost a doubling of the above ground biomass of a single tree, codominant in these stands after about 80 years. Again, these are preliminary data, and we're going to flesh this out by adding a lot of replicates. But you may be asking, well, what about variability within ecosites? So we do have some ecosites in our first year of sampling where we have quite a bit of replication. So here's 4 different traces, all representing boreal ecosite 128. Okay, so these are all intermediate conifer swamps. They all have the same ecosite. If you look at the radial growth again over time, the growth signal is strongly conserved. It's pretty much the same in all 4 of these very different sites, but which have the same ecosite. And if we look at the biomass, then I won't go through basal area or diameter reconstruction, but if you look at biomass again, you do see that for the same ecosite, this does translate, even though there's not much variance there, into a difference in biomass. So our ultimate goal then is to take and have 10 or 15 sites per ecosite replicated, and to develop a standard curve. Now, unfortunately, you can't see the error bars on this, but this is total above ground biomass for codominant trees in ecosite 128, and this is a standard curve showing the development or the growth of that stand in terms of biomass. And what you can't see is that the variability around the mean is fairly high with only 4 plots right now, but when we add plots, that will decline and we'll generate more of these curves for each ecosite. I just want to finish up today by briefly mentioning some work that we've done with ground-based LiDAR. There's been a lot of interest in something called the value chain, and that is choosing where to divert wood products depending on the quality of the wood, either to timber, lumber, or biomass. And so, as Joe mentioned before, we can use allometric equations to try to determine what the balance of branch biomass to stem biomass is in these. And we may be able to add, using LiDAR, we might be able to look at some of the intertree variability by looking at stands of different density, different characters, etc. We also may be able to look at parameters of wood quality such as branch diameter and branch angle and use these to assess quality from a tree before having to actually take the tree down. So we started a collaborative study with Art Groot at the Canadian Wood Fibre Centre at the Wade Lake Uneven Age Silvicultural Experiment near Cochrane. And there are 4 residual density treatments at this site. We chose 5 trees from each density, and we did 4 scan locations of LiDAR for each tree. We measured a whole bunch of attributes, including diameter, height, and the angle and diameter of each branch on those trees. And then we've imported the data that we collected with the LiDAR into the lab, and we're creating a virtual 3D tree by examining the point cloud. Now, we're right at the beginning of this work, but I just wanted to show you that this is sort of what you can achieve with the ground-based LiDAR. This is a point cloud image of a single tree. So this is different than Murray's because it's taken from the ground instead of from the air. But you can look at detail in that stem, and you can calculate things like diameter anywhere in the stem, and we think that we might be able to get down and look at diameter of branches and angle of branches and examine those things which might affect wood quality. So in summary, by the end of this project, we want to be able to take a biomass recovery scenario like, let's say we have a stand of 10 hectares that's boreal ecosite 128 and age 80. What is the mean annual biomass increment? And how much of that biomass is allocated to branches, boles, and stumps? And what's unique about the project is that we're going to drive it all through relationships that are related to ecosite. Thanks very much. Hopefully I'm okay for time. And I'd just like to thank quickly all the people who've supported this project.